All right, we are going to talk about skeletal muscle contraction today. And before we actually get into the contractile processes, I want to go over a little bit of the anatomy of the skeletal muscle, just kind of refresh your memory, and especially even talk about the connective tissue, because it's very important in the movement of the muscle. And of course, you already know skeletal muscle is attached to bone, and so this is, of course, what's moving the body. All right? So we have an individual muscle cell here, all right? And the individual muscle cell is referred to as a myo, and myo would be for muscle. This is a myofiber. And skeletal muscle cells, if you remember, they are multinucleated. And one of the reasons for this multinucleated cell is because in the fetal stage of development, skeletal muscle cells are actually just single nuclei cells. However, as the fetus continues to develop, the single cells fuse together so that one muscle cell is as long as the entire muscle. And so you get this multinucleated cell. But now that also means not only does it have multinuclei, it has multimitochondria and Golgi apparatus and all that other organelle stuff all in that one muscle cell. So you have this very long cell. And that muscle cell is surrounded by a connective tissue layer, which is called the endomysium. So the endomysium surrounds this single muscle cell. Now the endomysium is a very sticky layer. So you almost think of it like fly paper, okay, that sticky stuff. And what it does is it helps to hold all of the muscle cells together, so that when these muscle cells contract, they all have the ability to contract in unison together. You don't want muscle cells contracting separately. It's not a lot of fun. This is what we would call a muscle twitch. You ever get that tick in your eye thing going on? By the way, you know, you get that tick in your eye thing going on, it just won't go away. That's a good sign of stress. You had that yesterday, huh? Yeah, so yeah, when you got to go, dang it, go away, that's usually a stress response going on. Now, muscle is also grouped in fascicles. So you have these bundles of muscle, and you can kind of see it really good here. These are the individual fascicles or bundles of muscle. And these bundles are surrounded by another connective tissue layer, which you can also see that connective tissue here. This is the paramysium surrounding each of these bundles or fascicles. Now, though that paramysium also helps to hold the muscle together, helps to stick it all together. And then on the outside of the muscle, holding all the bundles together, is the connective tissue layer called the epimysium. Remember, endo means inner, peri means somewhere around the middle, and then epi means on the outside. And the epimysium turns into the tendon, and you know tendons connect muscle to bone. So all of these connective tissue layers are going to become very important, and we'll talk about those as far as the ability for our muscles to contract. So in the next picture, what I'm going to do is we're going to take this individual muscle cell right here and just look at it and then all the things that it's made up of. So here's an individual muscle cell. This is the cell membrane, okay? And the cell membrane of a muscle cell is typically referred to as the sarcolemma. Now, just like myo, sarco has to do with muscle. So if you ever see myo or sarco as a prefix, you know they're talking about muscle. Lemma is used for cell membrane. So for instance, if I were to say to you, I have a neurolemma, what cell membrane am I talking about? On a neuron. If I want to be really generic, I can call it a plasma lemma, and this is just a cell membrane on any cell, okay? So this is our sarcolemma, and then you also know that muscle, for skeletal muscle, it is striated. And so we're going to talk about where do those stripes come from. And inside the muscle, you can't really see the organelles very well. That's because all of our skeletal muscle cells are just packed full of these long rods of proteins. And these long rods of proteins go the full length of this muscle cell. And those long rods of proteins are referred to as a myofibril. It's the myofibril that's going to do all the heavy lifting, you might say. It's the myofibril that does the contraction that allows our muscle cells to move. So we need to spend a little bit of time talking about the myofibril. So that's what we're looking at here. So what we've done is we've taken this individual muscle cell right up here and the myofibril and pulled the myofibril out to just look at it separately, okay? So this is one of those rods of proteins. Now, probably in anatomy, you had to memorize the I band and the A band and the M line and all that kind of stuff, yeah? Okay, so if we look back at this single muscle cell here with all the stripes, look at the individual rod of protein, and it is actually what is striped. And you can see those stripes through the cell membrane, okay? And those stripes all have to do, whoops, wrong picture, all have to do with this I band and A band and M line and everything. So there's the light area and the dark area and the light and the dark, and they keep repeating light, dark, light, dark. And that's because we have this, sorry ghost, you have to go. We have this repeating pattern on these rods of proteins, or this myofibril, and this repeating pattern is bounded by these two Z lines, or you might have learned them in anatomy called a Z disc. It's the same thing, doesn't really matter. And this is what we refer to as the sarcomere. Down the center of this sarcomere, you also have something referred to as the M line. Now, the Z lines and the M line are actually proteins, and they're what we would call like anchoring proteins, things attached to these proteins. Okay, now for physio, I don't really want you to go back and rememorize I band and A band and all that kind of stuff. All you need to know are Z lines and M line. Z for the end, M stands for the middle. So you got the middle lines and the M, or for the M line, and the Z lines are the end lines. So if you look at this picture here, you'll see this is the Z line here, kind of in blue, another Z line. And then this dark line right here, this is the M line. So from Z line to Z line, with the M line in the middle, this is one sarcomere. Okay? And this repeats over and over again down the length of our muscle cell. So the Z line is where the light area is. So you can see Z line, Z line, Z line, Z line, all the way down. The sarcomere keeps repeating all the way down this muscle cell. Now, the Z line and the M line, like I told you, these are anchoring proteins, things attached to these proteins. And the things that attach to them, you've heard of these before, we have what are called filaments. And simply put, the filaments are a type of protein. And you may have heard of the thick filament and the thin filament. And the thick filament's name is myosin, and the thin filament is actin. Ring a bell? Okay. So looking at this picture here at the bottom, you can see we have the Z line, and then we have the M line. Myosin is in red here, and it is attached to the M line. It is the thick filament. So that's what we're going to have here. This is going to be our myosin attached to the M line. Actin, on the other hand, which in this picture is actually the blue lines, actin is attached to the Z lines. So our thin filament is attached to the Z lines. Now keep in mind that what we're drawing on the board is just this two-dimensional picture, whereas muscle is 3D, okay? So if we were looking at the myofiber, this is the actual rod of muscle that we're looking at, we might have something like this, where this is the myosin, okay? And this is the myosin rod, and then the actin is not just above and below like I just drew it for you. The actin is actually all the way around this myosin rod. So you'd have about 5 actin all the way around. So if this was the myosin rod, you'd have actin above and below, out towards you, back towards me, and so on. Okay? It goes all the way around. Any questions so far? Okay, so this is just a little bit better picture showing you the Z line with the myosin attached to the M line, the actin attached to the Z line. Now myosin, if you remember, this thick filament is composed of this kind of protein that has this sort of long twisting tail, and it looks like maybe 2 golf clubs that were wound around each other, and then you have the 2 heads. But to make it really thick, you take a bunch of these golf clubs wound around each other and you stick them all together. And always kind of reminds me of, you know, like those long cheese logs you see at the stores at Christmas time with all the nuts coming out the side. Anyway, that's my way of thinking about it. So you've got all these myosin heads protruding. Now, in the resting state of our skeletal muscle, all of the myosin heads are pointing towards the nearest Z line. So although that picture makes it look like they're all over the place, they're actually pointing towards their nearest Z line. Actin is a little bit more complicated than the myosin. Actin is actually made up of several different proteins. So one of the proteins that makes up actin is this kind of globular structure thing that somebody said kind of looks like 2 strands of pearls sort of wound around each other. So these globs sort of look like pearls in 2 strands is part of our actin protein. And since these are globs of protein, these have been named G-actin, and the G stands for globular. So this is globular actin. So one of the proteins that's involved in our actin that's attached to the Z line is the globular actin. one of the important things about globular actin is that there is a binding site on each of these globs of protein, each of these little pearl-like structures. And this is a binding site for the myosin, especially the head, to be able to bind to. So on this G-actin, we have a binding site for the myosin head. And remember, the way that this is set up, the myosin heads are protruding in all directions out of the myosin towards the nearest actin. And that's all in 3D. So you'd have here, these in purple are all your actin, and you'd have myosin heads coming out in all directions here towards that actin to be able to bind to it. So we'll just say this is the myosin, and of course you know that these are the myosin heads being able to bond to the G-actin. Don't laugh at my drawings, I know what you're thinking. Everybody thinks that every time I draw it. That's not the myosin bud, it's the myosin head, okay? You feel that? Not really. I know my drawings suck. All right, now, here's an important thing for you to know. The myosin has a super high affinity for that G-actin. They want to be able to bond to each other. And if the myosin head can bond to that G-actin, muscle contraction will occur. So let me say that again. If this myosin head is able to actually bond to the G-actin, and it really wants to, they have a super high affinity, if the myosin head can actually bond to that G-actin, muscle contraction will occur. Now they sit right next to each other all the time. So this could potentially be a problem because since myosin and actin are right next to each other, they have a super high affinity for each other, they always want to bond to each other, which means that we'd be in constant tetany. Tetany means our muscles would all be super contracted all the time. This is bad. So if you pretend this is actin and myosin bonding to each other, what we need to do is we need to be able to separate the two so they can't get to each other. And there actually is a protein that can keep the two of them from bonding to each other so that we have the ability to not contract, so we have the ability to relax. And there's this kind of ribbon-like protein that literally can separate these guys from each other for the relaxation process. This ribbon-like protein is called tropomyosin. So if I have tropomyosin in between my myosin head and my G-actin, then I can have relaxation of my muscles. So again, if you can imagine this tissue is my tropomyosin, if I put this in between my actin and myosin, the two can't actually bond to each other, and so when it's in between, my muscles will be relaxed. However, if I remove the tropomyosin, and now my actin and myosin can actually bond to each other, I'll have contraction of my skeletal muscle. So in this state, with the tropomyosin in between, muscle would be relaxed. If I could take this tropomyosin and pull it out and away, myosin and actin would bond, muscle would contract. Question?
That's what happens with tetany. Is what happens with the tropomyosin?
Tetany means tropomyosin has been removed and muscle just keeps contracting and contracting, and there's no way to put the two back.
So is that what happens when you're low calcium and you have tetany? It's the same. It's tetany.
Well, hold that question. We'll get to that. I'm going to answer that for you. But I need to tell you a few things first. So obviously in the healthy state, we want to be able to relax muscle and have the tropomyosin in between the actin and myosin. And sometimes we want to pull away the tropomyosin so the actin and myosin can bond and I can contract. So we have to have something that literally pulls that tropomyosin out from in between actin and myosin. So attached to the tropomyosin is another protein. This protein is called troponin. And if you can kind of think of troponin like a handle, troponin has the ability to move tropomyosin back and forth. So tropomyosin either slides in between actin and myosin and muscle relaxes, or troponin pulls the tropomyosin away so the actin and myosin can bond and muscle contracts. So troponin is literally the handle that's moving tropomyosin in between or out for relaxation, contraction. Any questions so far? So if I want to contract this muscle, I'm going to have troponin swing tropomyosin out from in between actin and myosin. Now the actin and myosin can bond, muscle will contract. If I want relaxation, I'm going to have troponin swing tropomyosin back into place so that it's in between myosin and actin and they can't bond and muscle will relax. The next question, so go ahead.
So it doesn't, it just spins the tropomyosin.
Spins?
Like it rotates it around.
It pulls it out from in between.
Okay. Because I was thinking like of the 3D portion of it.
Okay.
And
so it would be right here. So it's going to pull it out from in between. So you have different areas of tropomyosin. Any other questions? Okay, so now the next question is, how does troponin know when to remove tropomyosin? And how it knows is calcium is involved. So we're going to have calcium come in and bind to our troponin. And calcium causes contraction. If calcium is present, muscle will contract. So if calcium binds to troponin, troponin is going to change shape and remove tropomyosin so that it's not between actin and myosin anymore, and the two of them can bind, muscle will contract. If I remove calcium off of troponin, troponin will swing around and put tropomyosin back in between actin and myosin, the muscle will relax. So anytime calcium is present on troponin, it causes muscle contraction. If calcium is not present, we have muscle relaxation. All right, so we have been talking about calcium binding to troponin and causing troponin to change shape. Remove that tropomyosin. Now actin and myosin can bond, and you have muscle contraction. The question is, where's the calcium coming from, and how does it know when to bind to troponin and when not to bind to troponin? So let's look at the overhead just for a second. And in this picture, you can see right here is an actual skeletal muscle or a myofiber. And then these are the myofibrils or the rods of protein that contain the actin and myosin. So this is a picture of those rods of protein, just blown up so we can see it better. Here's our Z line and our M line, and the myosin is attached to the M line, the actin to the Z line. Now, because there's not a lot of room inside skeletal muscle cells, the organelles have to become a little tricky in order to fit. This, right here in this kind of bluish purple color, this is the rough endoplasmic reticulum. And what it's done is it's wound itself around, kind of looks like a little spider web type of thing, and it's around each of these myofibrils or these rods of protein. And you know that the rough endoplasmic reticulum has two major functions. Function number one, it helps in protein synthesis, and function number two, it stores calcium. So the calcium that we're going to need to bind to troponin is inside this rough endoplasmic reticulum. Now, to let us know that, to let us know that this rough endoplasmic reticulum is in a muscle, we're actually going to call it the Sarcoplasmic Reticulum. Okay? So the Sarcoplasmic Reticulum is the rough endoplasmic reticulum. So in the picture I've drawn here, this line represents the cell membrane. And this cell membrane of our skeletal muscle is coming into close proximity with a motor neuron. And this motor neuron has within it a neurotransmitter, which we call acetylcholine. Acetylcholine is the neurotransmitter that will stimulate skeletal muscle contraction. And underneath the surface of this cell membrane is the rough endoplasmic reticulum. And then if you could imagine taking this picture and just putting it over top of this picture here. So our rough endoplasmic reticulum or sarcoplasmic reticulum sits right over top of all of our actin and myosin. Where our terminal boutons of our motor neuron come into close proximity to our muscle cell, this is not referred to as a synapse. Remember, synapses are only where neurons talk to neurons. Instead, this is the neuromuscular junction. So that's this region right here where the two of them are communicating with each other. This is our neuromuscular junction. Now sometimes you'll hear people also refer to this region as a motor end plate. But we'll really call it the neuromuscular junction as we're talking about it. Alright, so now skeletal muscle contraction is voluntary. So I need to just think about it to get my muscles to contract if everything's nice and healthy in my body. So maybe I want to just move my finger. So the first thing that's going to happen is I'm going to start thinking about, okay, I want to move my finger, and eventually I'm going to get action potentials going down the motor neuron. And those action potentials are going to cause the release of acetylcholine into this cleft here. Now, it's not the synaptic cleft, so we're going to call this the neuromuscular cleft. And I'm going to have exocytosis of acetylcholine, and my acetylcholine is going to bind to these receptors on the surface of my skeletal muscle. Now, because acetylcholine is binding to these receptors, we refer to these as cholinergic receptors. Now I want you to know the name of these receptors because there's a lot of different medications that you'll introduce to your patients which are cholinergic medications. So some of these medications will stimulate these receptors, some of these medications will inhibit these receptors. So you'll have what are called cholinergic medications, or they might be anti-cholinergic medications. Okay? So again, I'm thinking about moving my finger and I'm sending action potentials down the motor neuron, and the motor neuron is releasing acetylcholine into the neuromuscular cleft, and it's binding to the cholinergic receptors. Now the interesting thing about muscle is that they are very similar to neurons. All of the cells of our body have a resting membrane potential, high sodium concentration on the outside of the cell, low sodium inside, high potassium inside, low potassium outside. Same thing with skeletal muscle cells. But skeletal muscle cells, like several other cells we'll talk about, can also have action potentials. So they have voltage-regulated gates, and they have sodium-potassium pumps, and all the stuff we learned about the neuron can happen across the surface of a muscle cell too. So that when this acetylcholine binds, we actually see action potentials beginning to move across the surface of our skeletal muscle. Now you know, as action potentials are formed, one of the things that action potentials do is they repel potassium. And potassium is going to accumulate and is going to start the next action potential, which will then repel potassium. And that potassium accumulates and starts the next action potential. And the action potentials are spreading in both directions across the cell membrane of our skeletal muscle. However, what we really care about is the fact that underneath the surface of the cell muscle, there are these open tubes. And it's kind of like if you could think about, you're walking along not paying any attention and don't realize that there's an open manhole in front of you, and you just keep walking and down you go. Same thing's happening here with potassium. As potassium is being repelled across the surface of this membrane, there's an open manhole here, and some of this potassium is going to fall through this open manhole and fall into what we call the transverse tubules, or sometimes they're just referred to as the TE tubules. And we'll see an accumulation of potassium inside our transverse tubules. So one more time from the top. I'm thinking about moving my finger, and I'm sending action potentials down my motor neuron. And my motor neuron is going to release acetylcholine into my neuromuscular cleft. And the acetylcholine is binding to my cholinergic receptors, and this starts action potentials. And the action potentials are spreading in both directions across the surface of my skeletal muscle membrane and repelling potassium. Some of that potassium is going to fall into the open transverse tubule and start to accumulate. Now, resting membrane potential for skeletal muscle, just like a neuron, is minus 70 millivolts. What happens to my membrane potential if potassium starts to accumulate in any one place?
It's going to rise.
It's going to go up. Exactly. So notice here, my potassium is accumulating. And eventually, I'm going to reach minus 50 millivolts in my transverse tubules. What's the voltage by which calcium voltage-regulated gates open? Minus 50 millivolts. Very good. So on the sarcoplasmic reticulum, we have a bunch of calcium voltage-regulated gates, which when the transverse tubules, and by the way, all of them are accumulating potassium here, when they all reach minus 50 millivolts, it will cause the calcium voltage-regulated gates to pop open. And now my calcium can diffuse out and bind to my troponin, which is right underneath my sarcoplasmic reticulum. The calcium will change the shape of troponin. Troponin will remove tropomyosin from in between actin and myosin so that those two can bind, and I have skeletal muscle contraction.
So the calcium, I know you said that the sarcoplasmic reticulum is on top of the myosin, correct?
Yes.
So it'll just flow freely through something to get to it? Or
Well,
how does
the something it's going to float freely through is the intracellular fluid.
oh,
But notice, okay, again, in this picture, let's go back to this. Here's my sarcoplasmic reticulum, right? And it is directly on top of my troponin. So when calcium is released, it doesn't have very far to go. And it's going to have an affinity for troponin, so it's going to bind. And this is packed with all of these myofibrils. So there's really no place for calcium to go. It's not going to be able to freely float away someplace. It's going to be attracted right to troponin.
If you don't mind, please repeating after the calcium is accumulated in the tubules. From there, it opens up, what? Sorry. I missed that.
Too bad. No, I'm just joking. Let's do the whole thing from the beginning, okay? All right. So, I want to move my finger, okay? So, all about voluntary. And I'm going to activate the motor neuron. So, I get action potentials coming down the motor neuron. I'm releasing acetylcholine. The acetylcholine is binding to the cholinergic receptors and stimulating action potentials which move across the membrane. Now, the big deal about that is it's also repelling potassium, which potassium falls into my transverse tubules and starts to accumulate. I'm going to change the voltage from minus 70 millivolts, which it was at rest, but because my potassium is accumulating, I'm going to hit minus 50 millivolts, which, as you said, is the voltage to open all these calcium voltage regulated gates on my sarcoplasmic reticulum. And now calcium can just freely diffuse out and bond to my troponin. And now, of course, when calcium bonds to troponin, it removes the tropomyosin, actin and myosin bond. We're going to have muscle contraction. Got it? Awesome.
Is that why a lot of large bodybuilders, or would they have a large or a high potassium in the blood because too much of that potassium is being released?
They should not. No, but think about this also, okay? You have a bodybuilder, right? And what is bodybuilding all about? Okay, well, they're lifting weights. Hopefully we're doing this legally without steroids, okay? Not likely, but we'll talk about that anyway in a second. And so they're lifting weights, okay? And what is lifting weights going to do to skeletal muscle? Well, this is interesting. Anytime we do anything anti-gravity, I pick up something that's a little bit heavy, doesn't even matter if it's tiny bit heavy or a little tiny bit heavier, or if it's super heavy. Any anti-gravity type of movement that I do actually stimulates, and nobody's exactly sure how, okay, but it stimulates protein synthesis in skeletal muscle. So any anti-gravity types of things I do stimulates proteins to be built in my skeletal muscles. So, you know, some of you may have gone to places like Gold's Gym or whatever, someplace, and they tell you, you know, when you work out, how do your muscles get bigger? What do they tell you?
They like tear?
They have to, exactly, they have to rip and they have to tear. And when they rip and tear, they grow back bigger and badder and stronger. I don't know about you, but torn muscle does not sound good to me. Just doesn't sound right. Now, there is some destruction of protein when we really are lifting weights and we are really kind of abusing the muscle a little bit, okay? Some protein does break down. We do have to rebuild it. However, it's not a ripping and tearing of actual muscle. We got to get down to the cellular level here of what's actually going on. When we do weightlifting or anti-gravity types of movements, at the cellular level, we are building more actin and myosin. So if you can imagine each individual cell building more actin and myosin, that cell is going to get bigger. And so the reason that our muscles as a whole get bigger is because we've built more protein inside each cell. Do they still sell ballpark hot dogs? They used to have a commercial. You cook these, they plump when you cook them. Okay, that's kind of what your skeletal muscle cells do. They plump when you use them. They get bigger. And you don't really get the ripping, tearing effect. That doesn't truly happen. Now, steroids, the reason that a lot of people take steroid hormones, especially testosterone, is because testosterone also stimulates protein synthesis. And if you take a lot of testosterone, and then what will happen is it stimulates protein synthesis at a very high rate. And so you make more actin and myosin. The more actin and myosin you have in each individual muscle cell, the stronger the cell, the more you can lift up. So if you're trying to get your cells to lift up a certain weight, they're going to grow more actin and myosin so you can do it. Now, the reason people have to take steroid hormones is because we all have a genetic size that we're able to get to. Okay? And you've seen some guys, I mean, they've taken no steroids whatsoever, and they're huge men. And then you've seen some men who, they're just really small men. Okay? So it really has to do with genetics, and we can only all build our muscles to a certain genetic size. And some people are definitely going to be able to have a larger genetic size than others. But I can defeat genetics by taking testosterone. And I can force my body to build bigger muscles. The only problem is testosterone also doesn't only increase protein synthesis, but it increases mitosis. And anytime we increase the mitotic rate in the body, we are increasing our susceptibility to cancer. Now, there is a disease, before we go on, that I want you to know about. I think they took the H out of it now. That's the old way to spell it. Myasthenia gravis. This is an autoimmune disease where the person's own immune system starts to attack and destroy their cholinergic receptors. So that these receptors, over time, don't exist anymore. So what's going to happen if these receptors start to go away? You can't contract your muscles. So one of the symptoms that you see in your patients is they come in, and a really common one is like the eyelids are drooping. They can't barely open their eyes. They might come in and they're very slumped down as they walk in, or they might sit down in a chair and just do this because they have no energy to keep posture, to keep their head up. I shouldn't even say energy. They don't have the ability to bind acetylcholine. They can't make their muscles contract. I'll show you a video when we're done of this, and then the medications that are used to help to prevent some of this problem. So everything that we just talked about, this whole process, is referred to as excitation, doing with the neuron, contraction, coupling. So for your exam 3, this is the first half of this essay. So you're going to explain a little bit about the sarcomere, Z lines, M line, actin, myosin, what they're attached to, where they're at. And then you're going to explain a little bit about the structure of myosin, what it looks like, and the 3 major proteins that make up actin. And then you're going to talk about the motor neuron and the action potentials releasing acetylcholine, and tell me how calcium is released and binds to troponin. And then you're going to tell me, once calcium binds to troponin, it removes tropomyosin so that actin and myosin can bind. And once you tell me about actin and myosin binding, then you're going to talk about the second part, which is the sliding filament theory. So that once actin and myosin bind, we're going to get myosin to move actin. Okay? So we have to actually get the myosin heads moving the actin. So remember I told you that these myosin heads are pointed towards their nearest Z line, okay? And now the myosin and actin are going to bond to each other. So this myosin might bond to an actin above, it might bond to an actin out here. Wherever there's an actin, it's going to bond. And when we get myosin to move, it's going to take the actin and pull it in this direction towards the M line. And these myosin heads are going to grab a hold of their actin and pull it in this direction towards the M line. So that the Z lines are being pulled in towards the M line. And our sarcomere is getting shorter, it's getting smaller. Now imagine if I have that one long rod of protein. And every single one of the Z lines are being pulled in towards the M line. What happens to the size of this rod of protein? It gets shorter. But what's also going to happen is it's going to bulge in the middle as it shortens. Okay? Now mine doesn't do much bulging, but you get the idea. When you move a muscle, it's supposed to bulge in the middle. And that's because all of those sarcomeres are shortening as we contract this muscle inward, which of course you know means that the muscle is pulling on the bone. So we're going to talk about what we refer to as the sliding filament theory. And this is actually the shortening of this myofibril, or the sarcomere, in order for muscle contraction to occur. There are 4 steps to the sliding filament theory. The first step is where myosin and actin actually bond to each other. Okay? So when myosin and the G-actin globules, when they bond to each other, we form what's referred to as a cross bridge. Now I also want you to know that the myosin head, in order to bond to G-actin, has to be in a very particular shape. And keep in mind this is a protein, so it has the ability to change shape. The shape that it's in is called the high energy shape. So when the myosin head is able to bond to the G-actin, it is actually in the high energy shape. The last thing I want you to know about this step is that already attached to the myosin head is an ADP and a phosphate. So the first step is what we refer to as the formation of a cross bridge. And this is when the myosin head is in the high energy shape. It has an ADP and a phosphate attached to it. And it bonds to G-actin. As soon as actin and myosin bond, we're going to go to the second step, which is referred to as the power stroke. So in this case, the myosin head is going to change shape and pull on the actin. You can almost think of it like you're playing tug of war, okay? And so you're going to go play a game of tug of war against a different team, and your team all picks up the rope and grabs a hold of it. When you first grab a hold of that rope, your hands are in a certain position. You got a hold of the rope. So if you can think of the rope like the like the actin, and your hands are the myosin heads. And the whistle blows or whatever, and you're ready to play tug of war. Now there's one rule in this tug of war. You can't move your feet. So how are you going to win?
Use your back.
Yeah. Well, you're going to move your hands, right? You can't move your feet. You're not allowed to walk backwards. That's the one rule. So I grab a hold of the rope, and now I'm going to pull, right? And what I just did is I changed the position. I changed the shape of my hands. So if my hands are the myosin head holding onto the actin, the power stroke is the pull. And so myosin is changing its position, changing its shape to be able to pull on that actin. And literally when it's pulling on that actin, it's pulling the Z line towards the M line so that it can shorten that sarcomere. Now, of course, you know, when you play tug of war and you pull that rope, did you just win pulling it the first time?
No.
Oh, heck no. You don't win because they're pulling also. Okay? So keep that in mind because we'll go back to that. One other thing that happened when that myosin changed shape and pulled on the actin, the ADP and phosphate fell off. So in step one, playing this tug of war, myosin is in the high energy shape, it's pumped, it's ready to go, and it grabs a hold of the actin rope. Of course, that ADP and phosphate is attached as well. And then in the second step, we got the power stroke where we're going to pull on that rope. The myosin is pulling on the actin by changing its shape, and the Z line is getting pulled towards the M line. The ADP and phosphate falls off. However, with one pull, that's not enough to shorten your muscle all the way. It just shortens your muscle a little tiny bit. So if the rule of the tug of war is you can't move your feet and you pull, how do you suggest you win? What would you do next? Say it again. Oh, sorry, there's another rule. You can't do hand over hand. Sorry, I forgot to tell you that rule. Can't move your feet, can't do hand over hand. So you just pulled, how do you win? Say it again. Get more players. What are they going to do?
Pull behind you.
Pull. Now, if they're pulling, should you keep holding that rope?
No.
Heck no, you can get rope burn because you can't move your feet. So you probably should let go. If the guy behind you is pulling, let go, right? Because if you let go, could you grab again?
No.
And pull again, right?
No.
So this is exactly how muscle works. The guy behind you is pulling, you let go. So that you have the ability to pull again. Somebody behind you pulls while you let go. And you pull again. The other guy behind you pulls. Teamwork. And you pull again. So what we have to do now is you just pulled. So depend on your team members to still hold the rope, but you're going to let go. And that's the third step. It's actually myosin lets go of actin. Now, in order to let go, though, myosin has to totally change shape in this step. It has to now go to what we call the low energy shape. And, of course, one of the ways we know to change the shape of a protein is add a phosphate. So along comes an ATP. And it's going to bind to the myosin head, and that's what causes it to change shape. So ATP comes along and binds to the myosin head, causes the myosin head to go into the low energy shape. And in the low energy shape, the myosin head lets go of the G-actin. We just let go of the rope.
So, like, if it's in the high energy shape, and someone like overextends their muscle, is that how like pull a muscle where it goes against contraction, and
Yes.
you can sprain the muscle?
Yes. In overextending a muscle, you can rip the muscle, you can rip connective tissue. Okay, so now what we've done is we've grabbed a hold of the rope and we've pulled, and now we've let go of the rope. So we got to go back to grabbing a hold of the rope. However, myosin's in the low energy shape, and it has to be in the high energy shape to grab the rope. So we got to get rid of the ATP that's on the myosin head and get it back into the high energy shape so it can grab the rope again. Located on the myosin head all the time, it's always there, there is an enzyme. This is ATPase. It lives on the myosin head all the time. ATPase has the ability to break down ATP. So the ATPase breaks down the ATP into an ADP and a phosphate. And this allows the myosin head to go back into the high energy shape, which means we can go back to step 1 and form a cross bridge and do the whole thing over and over again. And by the way, this is where that original ADP and phosphate came from in step 1.
Okay, so then people get fatigue or weakness, muscle weakness, when they're having issues building ATP and in their cells, like nutrition-wise. So the whole system shuts down everywhere.
If I can't make enough ATP? Oh, sure.
Okay, and that's the first thing that happens is towards your muscles.
No. If I start to get fatigued due to
Lack of
using my muscles, usually that fatigue is central nervous system fatigue first. So my central nervous system, my thought process of doing this usually gives up first. That's why your coach is screaming at you, you can do it, you can do it, because they don't want you to fatigue in the central nervous system. A lot of it is, I don't think I can do it anymore, and so I give up. Okay? However, sometimes there is actual muscle fatigue, and that could be from a number of reasons. One is definitely nutrition. Maybe I'm not eating right, so I'm not making enough ATP. Maybe I don't have enough calcium. Maybe I have sodium, potassium issues. I got to be able to have neurons function. I have to be able to have my muscle function. So it can be a whole host of different issues that create muscle fatigue. But most of it, you're correct, is due to lack of ATP. Now, if I'm an Olympic athlete, it's probably not ATP, unless we're kind of like the American Olympic athletes. If you read what they eat for breakfast, lunch, and dinner, it's ridiculous.
Yeah, it's like a bunch of pasta and
They're
bread.
eating McDonald's and candy bars and crap like you would not believe. What's the name of the guy that won all those swimming records?
Michael Phelps.
Okay.
Yeah.
Read his nutrition journal, and it's amazing the guy could do anything at all. He is a junk food junkie. And of course, it's showing now he has multiple different autoimmune diseases. And it's really hurting him because he's beat up his body so hard to become this world-class athlete, but he never gave it the correct nutrition. And now his body is fighting back and saying, nope, this is what you get. So you have to be really careful when you decide you're going to work out and work out hard and continue to eat the way that you used to. The body does not appreciate it. It has a tendency to fall apart.
Okay, and is that just like a spelling error? Is it Myosine?
Oh, I'm sorry. It's an,
I mean, I didn't know if it was like
used to be
changing
with
or
an E.
something.
Like potato used to have an E.
Change the spelling to make us more confused. They do.
That's right. I just want to screw you up and
No,
see if I
not
can-
you. Like,
Oh,
same with Myasthenia Gravis. She said there's no H.
there's no H anymore, yeah. Any other questions about this? Okay, one little last part. This calcium right here. It's out there. It's bonded to troponin. But what if I want to relax now? I've done all my contractions and I want to relax. What do I have to do? Well, I have to get rid of this calcium. Okay? And the first thing that's going to happen if I want to relax my muscle is I'm thinking about the fact I want to relax my muscle. Okay? And so I don't have a motor neuron being stimulated anymore. I'm not going to be releasing any acetylcholine. How do I get rid of acetylcholine that's already in that neuromuscular junction? Or neuromuscular cleft? How do I get rid of that neurotransmitter?
The three different ways.
There's 3 different ways. Enzymes are one. Phagocytosis, reuptake. Now, there's 3 different ways if it's a neuron, but there's only one if it's a neuron talking to a muscle, and that's enzymes. So only that enzymatic degradation thing works here. So we're going to have an enzyme chewing this neurotransmitter up, which means, as you know, all the action potentials stop. And then I have a pump on my sarcoplasmic reticulum. This pump is going to take calcium and actively transport it back into my sarcoplasmic reticulum off of my troponin. Now, by the way, this pump is called a calcium ATPase pump. It's like our sodium potassium ATPase. This is a calcium ATPase pump. And it can actively transport the calcium off of troponin back into the sarcoplasmic reticulum, where it came from. And now my troponin puts my tropomyosin back in between my actin and myosin. They can't bond anymore. My muscle relaxes. So as soon as I say, okay, I don't want to move my finger anymore, my brain stops action potentials down the motor neuron, and I get rid of acetylcholine with my enzyme. All the action potentials stop, and I have my calcium ATPase pump. And the calcium ATPase pump is going to pump my calcium back into the sarcoplasmic reticulum off of troponin. And now troponin will slip tropomyosin back in between actin and myosin. They can't bond anymore. By the way, that's the end of your essay. So you're going to talk about the sliding filament theory, and then you're going to tell me how the muscle relaxes by putting calcium back into the sarcoplasmic reticulum, and then your essay is over. Now, a couple other things, but not part of your essay, just kind of information. We talked about muscles getting bigger as we use them. We make more actin and myosin inside every single muscle cell. But I think I neglected to tell you that the more actin and myosin we have in every single muscle cell, the stronger our force of contraction can be. So the more actin and myosin that bind, the greater your force of contraction. Now the interesting thing is that when we pick something up that's heavy or not heavy or whatever it may be, our brain is trying to figure out how much actin and myosin should we have bond to pick that sucker up. We don't want to waste too much energy, so the brain sends certain action potentials down the motor neuron to say how many actin and myosin should bond. Because obviously if I pick this up, I don't want to have as many actin and myosin bonding in comparison to maybe picking this up, a few more actin and myosin bonding. But have you ever done this, where you see something that you think is really light, and you don't know that it's made out of lead, right? And you go to pick it up, and you can't. That's because the brain miscalculated. Or you see something that you think is really heavy, and you go to pick it up, and you practically fall on your butt because you've picked it up too hard, and again, the brain has miscalculated. Or maybe you've heard of, like, you know, that woman that weighs like 90 pounds soaking wet, and her kid is trapped in a fiery car after an accident, and she rips the door open, pulls the kid out, and throws the car like 1,000 feet down the road. The question is, could something like that happen? Well, maybe not quite that. But yes, we typically don't use all of our strength. So when we get into situations where we need to use more strength, after it's all over, you're like, dang, I didn't know I could do that. Because the brain doesn't normally want you to overuse what you have. It doesn't want to put you in those situations unless it knows you absolutely have to be in that situation. So really your brain is controlling how much strength or how much, you know, easiness of contraction it should be. And it's very interesting, and sometimes we don't really have voluntary control of that.
So in that case, when adrenaline starts kicking in...
When what kicks in? What do we call it? Epinephrine. Excellent. Go ahead.
When that kicks in, does that help you use complete muscle?
Yes.
Or?
Yes. Because what that's doing is that's sending different information out to the body to say, hey, look, use more acetylcholine, bind more actin and myosin. Yes, absolutely.
So why when you're weightlifting do they tell you like build up, not just like go and like hit the biggest? So why do they do that?
Okay. That is actually less about your actin and myosin than your connective tissue.
Oh, okay.
So all that connective tissue that we talked about in the beginning is very fragile. Connective tissue, like cartilage, has very little blood supply. Cartilage actually has 0 blood supply, which means that if you rip it and tear it, it's very hard to heal. And so they don't want you to start out with super duper heavy weights because, yeah, you could hurt the muscle, but muscle's got plenty of blood supply, it can heal. It's the connective tissue that does not like to heal. And all those layers of connective tissue have to hold all the different actin, myosin cells together so that when our cells start to move, it moves everybody in unison. So you don't get that twitching thing going on. So you want to be really careful about the joints, really careful about the connective tissue.
So the tendons farther down the line that are connected to the bone, that's the connective tissue you're talking about?
Well, that and also endomysium, paramysium, epimysium, all of that too.
Okay.
That can also rip.
Okay.
Yep.
Can you tell me what again, actin,
The three what?
the three major proteins that it's in
The G-actin, the tropomyosin, and the troponin. Okay, awesome.
So when someone has constant muscle twitching, like in a specific area, like for me, I have it in my left calf from a nerve problem.
Okay.
Does that also have to do with the calcium and the troponin constantly being stimulated over and over again, or would it be from a nerve problem?
Okay, so yes and yes. Because if you have a neurological problem with the motor neuron, it could be sending out flashes of acetylcholine, and then you get this quick contraction, and then it ends. So the motor neuron itself could be relating information to the muscle. It shouldn't, and you get a quick twitch. So definitely, if it's a neurological problem, that could definitely be happening. So you release calcium real quick, and then it stops, and you get these twitches that occur.
So then, for that specific area, would that also make it more deficient of the calcium or any of the other?
Not necessarily, not unless it's like constant. Okay, if it's constant, then yes, there is a possibility.
And then that's what would cause the cramping if it was
Yes.
out of the calcium.
Yes. Now, one other thing to bring up, and then I'll get to your question, Alex, that Greg kind of alluded to a little bit, is we all get constant input from the motor neuron, okay? Throughout our entire body, right now, every single one of your skeletal muscles is getting a little bit of acetylcholine being released constantly every second. So that every single one of our skeletal muscles are twitching very quickly. You can't feel it, but it's happening. Actually, that little burst of acetylcholine, they call it a quanta, a little packet of acetylcholine. Now, here's why this is important. Let's say you have a patient who is a quadriplegic. One of the things that we see from quadriplegics is that there's no neurological information going down the motor neurons, which means there's no quanta of electricity hitting their skeletal muscles all day long. And their skeletal muscles atrophy. They start to fall apart. So it appears that those little bits of stimulation of our skeletal muscles all the time actually keeps them healthy, keeps them producing proteins all day long. This is also why in the hospitals, especially like in physical therapy, they'll put on to your patients who are trying to build muscle, or if the muscle has been destroyed, they'll put this little electrical pack that shoots little bits of electricity into the muscle, and it helps to build that muscle. They call that the piezoelectric effect. And it's very, very helpful in producing muscle for patients who can't move around. Question?
So if I were to inhibit the enzymes that there are ACH, what would that do?
If we
What
had,
would be the consequence?
if we had something to inhibit the enzyme,
Yeah.
you'd be in tetany constantly.
What you're talking about, is that the same as the TENS unit, or is this?
That is a TENS unit.
Sorry.
No, go ahead.
So this pertains to smooth muscle as well?
Yes.
Yes?
The smooth muscle is very similar. There's a little bit of difference, but it's very similar. And we'll talk about this next week, but cardiac muscle is super duper similar.
So voluntary, but cardiac and smooth are not voluntary.
No, no, but we'll talk about the differences.
Okay.
Yep. So what happens if I don't produce ATP anymore? I have something called rigor mortis. And rigor mortis means stiffness after death. So if a person is deceased, they don't make ATP anymore, they're going to go into rigor mortis. Can you tell me how it is that all my muscles will tighten up after I die, and they'll become so stiff that if my hands were clenched, you couldn't get them open with a pry bar? Why does that happen? I'm telling you it's all due to a lack of ATP, but how does that all happen? Look at the steps.
So ATP can't be connected.
Does it have something to do with calcium? Yes, it does. Very good. So after I die, do I have a resting membrane potential anymore?
Mm-mm.
Nope, I sure don't. And usually my resting membrane potential is around minus 70 millivolts. And so as soon as we die, that resting membrane potential will go to 0. Now as it goes up towards 0, I pass minus 50, don't I? Which means that calcium gates pop open. And I start getting calcium flooding all of my cells throughout all of my body. And as it does, this causes the tropomyosin to be removed, and actin and myosin can bond. So it's as if we grab a hold of the rope and pull. But we can't let go of the rope. ATP is what causes the myosin to change shape and let go of the rope, but I don't make ATP anymore. So calcium causes the actin and myosin to bond, and the myosin pulls, and you contract, but the myosin can't let go.
So calcium's still attached to it.
Calcium's still attached because
How
there's
is
no
it still
pump.
pulling?
I'm sorry?
How is it still pulling?
It's
though?
not still pulling.
It
It
pulls.
grabs and pulls and then holds.
It doesn't release.
It doesn't release. So normally we grab, pull, release. Grab, pull, release. And one myosin is grabbing, pulling, and releasing while the other ones are holding. And then when this one's holding, the other ones are releasing. So we can keep doing that pulling thing. But after we die, we grab a hold and pull. And that's it. There's no release. Now, usually somebody goes into rigor mortis after they die, and we stay in rigor mortis for 24, maybe 36 hours, depending on the temperature. And then the rigor mortis starts to go away. And the body actually does unstiffen. But that's because now bacteria has started to take over, and it's eating away at the muscles, and it's causing the actin and myosin to start to fall apart, and now the body loosens. Which, for you, this is important for you to know, because when your patient dies, your job is to pose them. You will close their eyes, you will shut their mouths, you will move their arms and legs, and your job is to pose them. You will tie up body orifices and all kinds of stuff. Hey, you're picking this job, not me. I'm just saying, that's your job. Any other questions? Okay. Just want to make sure I didn't leave anything out. There was, oh, there it is there. I want to show you this picture because I thought it was kind of cool. Oops, hello. This is a picture of an electron micrograph, and this is a motor neuron. And this is a muscle fiber. And notice where the terminal bouton and the motor neuron, of the motor neuron and the muscle fiber meet. That's your motor end plate, or your neuromuscular junction. And then here's another one here, and so on. So every place there's a terminal bouton, it's hooking to this muscle fiber, which is kind of cool. One of the things we do know is that the more terminal boutons that attach to a muscle fiber, the better we have control of that muscle. So we have more fine motor movements. So for instance, if you look at a muscle fiber in the face, for every single muscle fiber, and this is not never mind. For every single muscle fiber, there's about 1,000 terminal boutons on a single muscle fiber. Whereas if you look at the gastrocnemius muscle, which you don't need it to smile or do hardly anything except for walk, there's 1 terminal bouton per muscle fiber on the gastrocnemius. So the better you need control of that muscle, the more terminal boutons you have. Kind of interesting.
I'll have to Botox your face.
They loose.
Exactly. All right. Looks good to me.